Today where you will learn about diffusion and osmosis. You will need to use these processes to save Frank's life. Click on Frank to learn about what happened to him.
Hi there. I stayed out in the sun too long and got very dehydrated. The doctor says I need a saline infusion, but I am not sure which bottle is the correct one. Do you have any questions? A. What is saline? B. What does hypotonic mean? C. What does hypertonic mean? D. What does isotonic mean? E. That's all. Let's continue. Normal saline is a solution of water and 0.9% salt. It is similar to blood plasma and can therefore be used in an infusion. Using pure water or more salt could damage the cells in the blood. Do you have any questions? A. A hypertonic solution contains more solutes or dissolved molecules than another solution, in this case, the blood. In other words, the hypertonic solution is more concentrated than the blood. Do you have any questions? A. Please take a look at the bottles in front of me and tell me what I should use for the infusion.
Hover over the bottles on the hand cart and select the one that should be used for the infusion. Let's not risk Frank's life. We better go to the lab first and learn about osmosis. Before we start, let's take a look at the difference between osmosis and another type of transport called diffusion. Click the Start Animation button on the left side of the screen. Which side of the container has more of the orange salt molecules at the end of the diffusion animation? The right side. B. Both sides have the same number. C. The left side. D. All orange molecules are gone. That's right. The molecules move spontaneously in the solution. If the concentration of molecules on one side of the container is higher than the other, more molecules will move from this more concentrated side of the solution towards the more dilute side than in the other direction. Diffusion is the spontaneous movement of particles from an area of higher concentration to an area of lower concentration. Think about how you can smell your grandma's delicious pie even from outside the kitchen. That is the diffusion of the odorous molecules traveling from a high concentration to a lower concentration. This spontaneous movement always happens and can go in any direction. When more molecules move from a higher concentration to a lower concentration in diffusion, we call this net movement, or net flux. When does the net flux of dissolved molecules stop? A. When the dissolved molecules are all on one side. B. When the concentration of dissolved molecules is equal on both sides. C. Never. D. When the amount of water is equal on both sides. That's right. The net flux stops, but the spontaneous movement keeps going. When the concentration is the same on both sides, an equal amount of dissolved molecules moves from left to right as from right to left within the container. Now compare this to osmosis by clicking the Start Animation button on the right. Osmosis is the movement of water molecules from a high water potential to a low water potential. High water potential means a lot of water, so a dilute solution. In which direction do dissolved molecules, also called solutes, move in osmosis? A. From low concentration to high concentration. B. From a high water potential to a low water potential. C. The solutes don't move. D. From high concentration to low concentration. That's right. In osmosis, the solutes don't move, for example, because they can't pass across a membrane. Instead, osmosis is the net movement of water from a high water potential to a low water potential until the water potential is equal on both sides. Now let's study osmosis by using some potato cells. Potatoes consist of cells just like your red and white blood cells. Cut the potato into 7 equally sized cubes with the scalpel. Pick up a potato cube. Measure the weight of the potato cube. Click the record button on the scale to record the measurement. Great! You made your first measurement of 2.78 grams and recorded it in the table on the screen. Click on the measurement on the wall screen. What does this value mean? A. The potato cube weighs 2.78 grams at 0% of the experiment. B. The potato cube that goes into the 0% salt solution weighs 2.78 grams. C. All of the potato cubes weigh 2.78 grams at the start of the experiment. D. The potato cube that goes into the 0% water solution weighs 2.78 grams. Cool beans, we are about to put this potato cube into the 0% salt solution. We need to weigh each cube before it goes into the solution so we can compare it to the weight of the same cube after it comes out. Now let's set up an experiment to see what happens when we expose the potato to a hypotonic solution. Put the potato cube in the most hypotonic solution. The potato cube is submerged in the solution. Solutes cannot move across the cell membrane, but thanks to small water channels, water can. Because of osmosis, the water will move across the membrane until the concentration of solutes is the same on both sides of the cell membrane. What's the salt concentration of pure water? A. 100%. B. 0.01%. C. 1%. D. 0%. That's right. This is pure water. By definition, there is no salt in it. That's why this is the most hypotonic solution. What do you think will happen to the potato cube? A. It will increase in weight. B. It will stay the same. C. It will decrease in weight. All right, let's see if you are right. I'll measure the weight of the other potato cubes and place them in the remaining solution so you don't need to do all that manual work. Take a look at the measurements and click on the screen. Remember when I told you to cut the potato into equally sized cubes? As a robot, seeing these different weights, it astonishes me how imprecise organic beings can be. Not a problem. We can correct for this. Let's leave the potatoes in the solution for a day. Pick up the potato cube from the solution with pure water. Blotting the potato cube removes water stuck to the outside of the potato cube before we weigh it. This is necessary because we want to measure how much water has gone into the cells, not how much is stuck to the outside. Remove excess water from the potato cube by blotting it on the absorbing paper. Now we need to measure the effect of keeping the potato in the hypotonic solution. Measure the final weight. Click the record button on the scale to send the measurement to the wall screen. Well done. I'll do the same for all the other cubes. You initially hypothesized that the potato would increase in weight when placed in a hypotonic solution. Indeed, it now weighs 2.9 grams compared to 2.78 grams at the start. You were right. This is science. Now let's calculate the percentage change. Click on the final weight of the 0% cube on the wall screen. In order to calculate the percentage change, we need to divide the weight change by the initial weight and multiply it by 100%. Now click on the initial weight to make the calculation. The difference between the initial weight and the final weight is 0.12. 0.12 divided by 2.78 times 100% equals 4.3%. How would you interpret this result? A. The weight has increased by 4.3 grams. B. The amount of salt in the potato has increased by 4.3%. C. The weight has decreased by 4.3%. D. The weight has increased by 4.3%. Exactly. In other words, the potato cube is 4.3% heavier after spending a day in the hypotonic solution. I'll calculate the percentage change of the other cubes as well and add them to the table on the screen. Click on the plot button on the screen to plot the measurements. Here we have calculated the percent change for all the cubes. Remember how the cubes were not exactly the same weight? By calculating the percent change, we correct for those small differences. Click the regression button to plot a linear regression. Click on the arrows on the chart to identify where a potato cube would not change in weight. You got it. This is called the isotonic point. The isotonic point is where the water potential inside the potato cube is equal to that of the solution it is in. Click on the arrow that points out the hypotonic solution. Why does the potato cube increase in weight in the hypotonic solution? A. Water moves from a high water potential in the solution to a low water potential in the cell. B. Water moves from a high water potential in the cell to a low water potential in the solution. C. There is a net movement of salt molecules into the potato. D. Water moves from a high salt concentration in the solution to a low salt concentration in the cell. That's right. The water potential of the solution is higher than that of the potato cells, so water flows into the cells. Which arrow points out a hypertonic solution? What describes a hypertonic solution? A. High salt concentration, high water potential. B. Low salt concentration, high water potential. C. Low salt concentration, low water potential. D. High salt concentration, low water potential. That's right. A hypertonic solution has a high concentration of solutes, in this case salt, and therefore a low water potential. Now let's see what would happen to Frank's blood cells if we gave him a hypotonic or hypertonic infusion. Take a cell culture plate. Place the cell culture plate on the microscope. Choose one solution and add it to the plate in the microscope. Click here for more information. The red blood cells shrink. Try adding the hypotonic solution to see what happens. The red blood cells are back to normal. Try adding more hypotonic solution. The red blood cells swell. Keep adding the hypotonic solution to see what happens. Oh no, the red blood cells exploded and died. As you can see, the red blood cells are permeable to water, just like the potato cells. When the salt concentration changes in the solution around the blood cells, water will move across the membrane until the water potential and solute concentration are equal on both sides of the membrane. When the water moves out of the cells, they shrink. When it moves into the cells, they swell and can even burst. Back in the hospital, you correctly suggested to use the isotonic solution for the infusion. Now you know why any of the other choices could have ended badly for Frank. Let me ask you one last question. Do you feel like you gained relevant knowledge during this simulation? A. Completely agree. B. Agree. C. Disagree. D. Completely disagree.